Morning everybody, my name's Toby Athersuch from Imperial College. Thanks very much for the introduction, also to the organisers for inviting me to this workshop. I'm pretty excited about coming to this, I'm not going to deny it, having followed the progress of these since over the first two workshops, and my involvement in some of the prototype exposome studies as they are. So my interest, I should probably say that I'm going to try and navigate a sort of narrow channel between what's been talked about already and what will come later in terms of the details of detection and separation. But I think I can do that and hopefully add some insight into why I'm interested in applying metabonomics to exposure science exposomics. So my interest really is in the field of environment and health, obviously, but my research group working at Imperial are working across a number of different aspects of implementing metabonomics in biological systems. So one part of this is the molecular phenotyping, which we've heard already in the previous talk, but also trying to link that back down to working on sort of in vitro and mechanistic studies to back up what we find in these other things. So I think one thing I would say out of the gate is it's very important that we actually do not forget that there are other things at play in trying to understand the role of exposures, and that we shouldn't just go whole hog into the metabolic phenotyping population studies in the absence of anything else. Because obviously when we add all these things together, we can use the mechanistic insight we gain from animal and in vitro models to inform us about the findings we find in populations and help move that forward to forming health, understanding of health, and also policy and action decisions. So in this talk I'm going to talk a little bit about exposome, some aspects that I thought I would highlight, although most have been already covered. I'm going to talk about continuum metabolism, and then some challenges really about what is difficult about the metabolome to measure, and why we're doing it and how we're doing it. And then I thought I'd round out by giving some examples from the recently developed MRC NIHR National Phenom Center, which sits at Imperial, headed by Professor Jeremy Nicholson, just show you what sort of facilities are available in the UK as a kind of flagship project. So we've got our environmental exposures coming from all over the show, and the challenge really here is that they are divided over many different dimensions. We've got a time dimension, which obviously people are exposed to things over different periods of time, and they may be exposed in different locations, different environments, with different periodicity. So I've put some up on here, but probably easiest to give some examples on this chart right here. So this figure is taken from a paper that I published a couple of years ago, and really there's maybe 5 different types of exposure we might look at. One might be an exposure that's in utero. Maybe the mother was smoking, and therefore the exposure was only for that short period and very little after that. Secondly, we might have something which is an exposure that accumulates in the body over time, perhaps a heavy metal exposure such as cadmium. Thirdly, we might have something which is an occupational exposure which only happens during the working lifetime. Fourth, we might have something which is maybe a xenobiotic exposure, maybe antibiotics or painkillers that's used at only certain periods of life, but maybe more at some than others. And lastly, we might have some kind of oscillating exposure, maybe even a biological, like something like an allergy or a response to an allergy, or even maybe taking a drug in response to an allergy on a very periodic basis. So given all this kind of complexity, I think it's just it would be worth to highlight that if we're trying to use population studies to get at which of these exposures are actually most important, then we really need to think about what sampling we're going to do in the context of those studies, given those types of exposures. So the two that really speak to this kind of prospectively collected study design are the EnviroGenome Markers project, which ran up to about 2 years ago and was really the prototype for doing these kinds of studies, and then more recently the exposomic study. There's also the helix study, which is based on mother-child cohorts, but it has less really to do with the meet-in-the-middle approach, which I'll explain now. So for those of you who have not come across this kind of diagram before, the idea is to take samples from people who were collected prior to them becoming sick and becoming clinically diagnosed with the disease, but we take them in a prospectively collected manner from a healthy population. And we select out those people who subsequently could become sick, and we match them with control individuals who don't, with many of the other confounding factors matched up. And then we can go back to the original samples we took from those people when they were healthy and try and identify which of the features in those samples predict their subsequent disease state, whether it's case or control. And the value, obviously, of these samples we can collect is that they are free from, well, largely free from the reactive type of pathways that Steve was talking about in his talk. And the idea is we can kind of get rid of those, offload those, and focus simply on those which reflect exposures or very early kinds of responses to those exposures which are intermediate in the development of the disease. So this is why it's kind of called this meet-in-the-middle approach, and this has been something that has been developed by Paolo Vinez at Imperial College and others. So, as I say, it's very important to select the right kinds of samples for this study. If we were going to try and use a cross-sectional study, then obviously it would have a very different impact on what we'd be able to say about the kinds of exposures that we are able to talk about. The other thing is to know about the sample time and obviously the time of life when we're selecting these samples, because obviously there are very different exposure profiles at each of these kind of critical periods of life, critical periods throughout life. So obviously if we sample only at one time point, then we're going to be limited in what we're able to attribute to the disease that we in our analyses. And so the second thing I'd really highlight is that although the studies we're currently using are prospectively collected in the majority, that actually one thing we really want to push forward is repeat collections in cohort samples. I think longitudinal sampling will be something else on top of what we're already doing that will allow us to chart people's progress through this sort of biochemical space and really allow us to understand which of those things are really giving rise to disease or reflect exposure or so on. Okay, so we're meeting in the middle. We're finding these kind of biomarkers that are predictive, or maybe they relate to exposure. And in terms of metabolite profiling, what we really need to do, obviously, make some kind of initial biomarker identification. And the power of what we're doing in metabolomics, the tools we use for detecting these kinds of things in the profiling are also the same we're able to use for structural identification of those same targets, those same metabolites, should we need to do that if they're not already known. We might also want to follow on by understanding in maybe an experimental study about the pharmacokinetics of these metabolites, and then obviously we need to go back and validate these responses in terms of this biomarker in other cohorts. But sitting at the centre of all of this is the analytics. So that's where we've got to today, and that's what I'm going to be mainly focusing on. So in terms of the things we're actually trying to measure, these metabolites, we've heard a lot about the different, maybe different definitions and all the different sources of metabolites that we can actually get, maybe the diet, maybe drugs, maybe from other external sources. But really these inhabit a kind of continuum from things you could consider to be truly endogenous and are necessary simply in the body, right the way through to those which could be considered truly xenobiotic and have no real function in terms of like normal biological function in humans. Then obviously there's this middle area, so we might want to define those other kinds of metabolites. And these speak to what some of the other speakers have said, in that we get feedback from some things, or we maybe have metabolism of some compounds which give rise to other things which have activity. So we can define these other types of metabolites, these xenobiotics, in terms of the xenometabolome that are in this space here. So the main point is here that we have a continuum, and that continuum really fills up a very large amount of chemical space. So we're thinking about now, okay, so now our problem is we're trying to cope with putting metabolomics in terms of the exposome, and we want to try and capture a broad range of metabolites across this entire continuum. Now, people in other omics have been saying, okay, fine, we can just profile everything. So what is the real problem with doing that in terms of metabolites? So I think the main thing is here, we can think of it in terms of codes. So bear with me. I've put up here a sort of dummy diagram of how we might consider some kind of genetic code. And we can interpret, say, DNA or maybe a gene transcript in terms of some combination of things within that code which give rise to some greater meaning to that code that it actually has in terms of biological function. And maybe even those things, so for example here we might have codons, and here we might have amino acids, and then the collection of these amino acids joined together might give us something that we can interpret. So we can interpret the original code, we can interpret what's happening here, and we can interpret this output. So the great thing about genomic information is basically down to measuring 4 different things a lot. We have the different bases, and we can measure those things. And that's why things have been able to ramp up very quickly from very simple kinds of genomic studies through to sequencing. Okay, because once you've got the technology to measure one of those 4 things very quickly, and you can do it a lot, then you just do it in multi-parallel measurements. And therefore, you can get genomes pumped out at a fantastic rate. So let's take another example. Again, bear with me. So maybe an epigenomic signature, maybe methylation of DNA at certain points. So here we maybe have the black dots are CPGs the white dots are CPGs sites where we could potentially have methylation, and these guys at the top are methylation of those sites. So quickly we can go through here, and we can whiz through our genome and say, okay, which of these sites do we actually have methylated? And again, it's kind of a binary thing. We're only measuring a small number of things, but we're doing it a lot, okay? So the actual analytics goes down to being able to do some small number of things a lot. Now, don't shout at me if you disagree, but that's I'm trying to make a contrast with the difference between that and what we have in terms of looking at metabolites. So here are 2 things which I've got from lipid maps, 2 phosphocholine metabolites, and they differ by basically only 2 carbons on one of these chains. The majority of this molecule is the same. Okay, we only know this is really different because of this very small thing at the end, and they may have very different biological consequences. The way we're reading this and the way we have to interact with samples that contain these 2 things, we want to differentiate them, go down to the physicochemical properties of these molecules. And as I say, in this case, that's relatively small. It's not simply a case of saying, okay, I've got my analytical equipment and I can chop this up, ding, 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 ding. Oh, there's 2 extra ones on the end of that. So going down to the very small differences in physicochemical properties between things becomes a massive challenge for understanding it. But my main point is that it's a chemistry problem in terms of trying to understand and evaluate what's in the metabolome. So as we've heard before, we've got a variety of different analytical techniques that we can apply, a whole different bunch of flavors of NMR spectroscopy, one-dimensional proton NMR spectroscopy being the main one. For profiling, it's quick, it's easy, and it's robust. It's non-destructive, and we can get out quite a lot of information in an agnostic fashion. We've heard also a lot about the different flavors of mass spectrometry we can apply, down for things which concentrate on getting very high resolution, so it's an FTMS, or maybe things which can give us a lot of information in a profiling manner very, very quickly, such as TOF-MS. So all of these things we can also hyphenate together with some kind of chromatography, and again we're going to hear about some of the challenges in doing this and separating out these things that may actually be very, very similar but may actually have importance in terms of us understanding what exactly are the metabolites of interest. And we can link all these things together with the automation as was discussed by David. Okay, so we've got these platforms that can generate these wonderful suites of metabolites, panels of metabolites, some in an untargeted manner. And we've seen already that when we put all these together, these are all the different techniques that we used in these two excellent papers which underpin these different metabolome databases, that actually we get complementarity between all these different types of techniques. So I'd say another challenge is one size of technique doesn't fit all. If you want to capture the metabolome, you actually need a lot of different techniques in concert. So it's not a case of saying, oh, I'm an MS kind of person, so I'll do that. Or I'm an NMR kind of guy, I'm going to do that. We really need all these things kind of work together to get as good a comprehensive coverage as we can, if that is in fact the goal of your study. Obviously that comes at a cost. It's quite relatively cheap to buy one mass spectrometer. It's not very cheap to buy 5 spectrometers all running in parallel with a NMR, or it takes a lot of time to do it all in sequence. Another thing is we need this because if we look actually at this plot that was shown earlier, where most of these things lie, about half of them are going to be visible by NMR, but the majority will be only visible by mass spectrometry kind of methods. So a challenge here is really the distribution of these kinds of molecules. And in terms of the exposome, these being 1,000 times or so, usually less abundant in our biosamples, then what can we really expect to find from each of these different techniques? So given this is true, and really these series of compounds here, largely the preserve of people doing analytical chemistry on a sort of individual or very small panel basis, what can we really do in terms of the analytics? What are we interested in putting together to look at both the things that aren't there in very high abundance and the things which we can probably routinely collect by most different types of methods? So obviously the things which are abundant, we can apply untargeted profiling methods. We're not needing to push the limits of detection by simply focusing on a small number of metabolites. And this is the thing that I'm most familiar with, and many others are. And I would say that this has the greatest opportunity to discover new things, especially in the within the concept of the exposome. There's things out there, we don't know what they are, we want to understand, or there's responses to those things, and we don't know what they are either. So broadening our view so that we can cover as much as possible in an untargeted way is probably going to be quite beneficial where possible. Has also the benefit of we don't need to have annotated all those things before, but as I said previously, the techniques we're using are also those we use to structurally elucidate these molecules, and then we can add them to these community databases. In terms of the other kinds of metabolites, the exposure metabolites, for example, say metabolites of PAHs or so, then maybe we do really need to go into targeted profiling of those specific molecules, and these kinds of targeted analyses have been included in these pilot exposome studies. Obviously, targeting, we don't need to just target those exposures, we can also target the responses, and as has been mentioned before, this gives a real opportunity to open up these kinds of metabolomic studies to the whole community. If you know what you're getting out of an assay, you say, I'm going to profile these 150 metabolites, they're going to come out, I know their quantities, and I can get on my way. You miss out, obviously, finding anything of novel metabolites, but you don't have to have a whole wealth of expertise in terms of annotating and interpreting untargeted spectra. And so, although I may have missed many, there are several vendors who are either doing services or providing kits for these kinds of analyses. So, in the interest of time, I think I'm going to skip past this because I've talked about study design. But for any particular study, we're obviously going to take some kind of collection. Here's an example where we might be collecting samples in a longitudinal manner from people over in the same place over many different periods. So, one thing I'd say is a real challenge for doing these kinds of exposome studies. They're usually done in a very distributed fashion across many different places, by different people at different times, and also getting different participants involved. And so coming down on a set of criteria for including samples that you now collect, or including samples that you have already collected, maybe before you knew what the exposome was 20 years ago, we can start to identify which of the sources of variation in terms of the collection are most important. And so in this paper we identified that some of the major contributing factors to what you can identify by LCMS, obviously anticoagulant, that's a chemical that you're adding to your sample, and it will interact, well, some of these anticoagulants will interact with the analytical hardware that you have. So that's not necessarily a problem as long as you know that that's something you need to account for. We found other kinds of factors. This was done on some freshly collected samples that were stored at different periods. So we found that, obviously the less time you leave something out on the bench, probably the more representative likely to be of the thing it started off being. But we found literally no real difference that we could interpret for samples that have been stored in appropriate conditions in -80 for very long periods of time. There appear to be no systematic differences with those. So if you want more information on this kind of thing in a practical sense, then I direct you to this paper by Hebels, which came out of the Envirogena Markers project. Okay. I've got some time. I'm good. Okay. So just wanted to say another issue that we have in terms of understanding when I've got a sample, when I've measured it, do I know whether that thing is any good or not? And here's another challenge we have. There is no real way that I'm aware of that can really tell you about the quality of a particular metabolite. Because a metabolite is a chemical. If it degrades and turns into another chemical, it's another chemical. So although you might be able to find ratios of things which might change, that could be one example, we don't have anything that's a very overt measure of quality like there exists for, say, RNA. You can get a RNA integrity number, which is done on an electrophoretic method, and basically tells you how good your collection of your mRNA is going to be. So this gives you an idea of whether your analysis is going to be any good or not in the end. We don't really have anything like that, and so we really need to put a lot of steps in place to understand if we can't know whether there's an intrinsic quality to these things, at least we can capture them in and preserve them in a state that's most representative of the sample we took at the beginning. Okay. Next up, some analytical challenges, which I'll present to the other speakers who are coming later. Here's a quick example, basically saying re-injections of a QC sample over many different injections, and we get one of these was done after a 2-day break, and we see separations in these QC samples as they're done by batch. The other thing is when we're running batches of samples, there may be analytical challenges there in terms of the instrument conditioning or instrument performance. So each of these 5 batches, you can see the initial QC had a very different response to subsequent QCs. So obviously you need to incorporate QCs as a major part of any of these kinds of metabolite analysis you're going to be running in exposome studies. Otherwise, if you have some of your actual samples being injected at this point into your run, then you get a very strange result because you're not, putting them into part of the run where things have become relatively stable. And lastly, I think probably the main community problem we're going to have is understanding the normal variation we expect. And the databases that have been compiled are going to be fantastically useful in this. But for any given study, understanding the biological variation we see in either these endogenous or xenobiotic compounds is something we really need to take into account. So this was a diagram generated from many spectra from rats in a toxicological study, but it gives you an idea that there's lots of different things that can influence where you would sit in this chemical space. And so understanding the normal variation we would expect to see in here gives us an idea about when we measure new things, whether we can really going to be able to say whether those things are something we can pursue in terms of their linkage to disease or exposure. Okay, so in terms of, I just wanted to mention one thing about actually practically doing this. So we may have metabolome profiles where we have xenobiotics compounds overtly in them. So NMR is very good at screening these out. And you think, okay, that's a good example, very useful for catching people who have not followed your study protocol. But likewise, these kinds of analyses that just draw out these things because they are different from a sort of a standard median profile would be a very good way of capturing maybe lifestyle or dietary components of the metabolome. So that's exactly what's been done in terms of identifying components of food which are represented here. There's another good reason to do this, and it's actually in terms of a practical thing, which is if you have an NMR and MS complementary metabolite profiling method, then you actually use the NMR first. So you can identify things that are going to be in your you can identify samples that may have gross contamination that if you took those forward into your LCMS study, then actually maybe you would find that it caused instrument problems with that. And that's what's being done in terms of the within the Phenom Center, which is the last part that I wanted to mention. So here we might have some samples, we screen them with NMR, we chuck out all the ones which have something very, very weird with them. They're unlikely to make it all the way through to the end of our analysis. Maybe they have some major contamination from their collection or some other factor, and they can be screened out and they won't affect the analytics later on. So for those of you who don't know, that's one of the procedures that's being incorporated into the high throughput analysis at the Phenom Center, which is, as I said, headed by Jeremy Nicholson, is in a partnership funded by the MRC NIHR in partnership with some instrument vendors. And it's basically NMR and mass spectrometry based analyses. And the main part of it really is, in terms of the exposome, we'll be looking to how we can measure all these different ways of getting the most information out of blood and urine samples. So they have quite a high capacity now, and I know there's similar endeavours going on at these metabolome cores in the US. So hopefully everybody can kind of work together and work out what the best way is to do this. But the way they're doing it is they've got 10 UPL CMS systems for untargeted kind of analysis, and then they've also got some targeted TQS systems for looking at individual biomarkers. And these slides are provided by the MS manager there at the Phenom Center, Matt Lewis, who basically illustrated that if you took your 10 UPL CMS machines, what are you going to do with them? So the first thing is really to divide them up into those that are going to run on a reverse phase kind of column and separate primarily things which have or hydrophobic in nature, and then you have a hydrophobic interaction chromatography, which will be much better at capturing and separating out polar compounds. So we've got the sort of two ends of the chemical spectrum there, and then you could divide the ones you've got for reverse phase into things that are in bloods and things that are in that are very lipidic species, and those things are more moderate, and then obviously things which are very basic. And you can divide these up in a number of different ways, and then prioritise some of these systems to only run in one particular ionization mode. So basically, if you have all these running in concert, you can take a study, large-scale epidemiological study, and basically pump in all the blood and all the urine to these different analyses. Okay. Obviously, behind all this comes the need to have fast and furious sample preparation, and so having optimised these kinds of procedures prior to this, you can then roboticize the whole thing, and using sample management systems get a very high throughput. So I think the main thing to take home from this is the profiling of large-scale epidemiology, you would imagine, obviously is not something that's going to be done by one guy on his own in a small lab. So I think the idea that was pushed earlier about having these community resources, having these go-to places where people can use these cores is an absolutely fantastic one, because there's no point to replicate all of this effort over and over again, developing all of these things, because ultimately if everybody develops things separately, then how are we ever going to mash it all together at the end and make some sense across these different population studies? Ultimately that's the basis. Right. So putting it all together, I don't need to go through all this. We've talked a lot about the types of things we can measure and why. We've got population studies which we can use in a prospective manner. We can define markers of biomarkers of exposure, those of response, and relate these two things together to understand how parts of these exposures we have here are actually related to those biomarkers of response. So, in conclusion, I will wrap up, and thank you for your patience. Firstly, untargeted metabolome tools are pretty well established, and I think the main thing there is that we need to think about what we're putting into these in terms of sample collection and those kind of caveats and pitfalls I talked about earlier. But also there's a greater need for sort of national and international collaboration to really use these databases, combine and make sure that we're using common protocols so we can combine the data that's being generated from these very precious sample resources. For targeted methods, I think the main part there is really that they open up metabolomics to a very wide audience. This really is something you can start doing on maybe smaller projects on your own in your lab, and therefore contribute to the sort of a community effort. But I would highlight the fact that they may be relatively limited in terms of finding novel exposures, as you might imagine. Obviously, combining the two has benefits in terms of coverage, but also in the fact that we can use NMR as a pre-screening tool to preserve the quality of the analysis we do in MS. And one final plea is that a lot of the studies that are done in this area may be coming from epidemiology and exposure science, who have the cohorts and the access to samples. Really encourage analysts you're going to be working with to come in and work with you right from the start to identify things like, have you got enough samples with the same anticoagulant, or do we have enough sample right at the beginning? Because finding that out later on can actually be quite a torturous process. Lastly, I'd say thank you to all those who support me and help me. People Jeremy Nicholson and Elaine Holmes, who run the department and my section respectively. Hector, who's been a big supporter and was the PI for the Exposomics project, the EnviroGenomarkers project on which I worked at Imperial. And also I wanted to mention those at the MPC who kindly I'm working with, and also have a student working with me now. So with that, I will finish, and I'll take any questions at the Q&A as well. So thank you. Well.